So I'm having a fundamental realization
I'll try to read it and see what whole blood on the side.
what that looks like. And then what I'll try is just to share the screen.
Yeah, and I mean, we need to seriously look at our cameras. Actually, James and Laura are also, they like my idea of us helping.
Yeah. So I think that should be it.
I'm frustrated with it.
And there's no
This whole situation that we get to.
indication that we're,
Teaching, now we're talking about. Because James did the same thing, he sent me an email, basically.
so can you log in from where you are?
Wanting to drop just with the task. Trying to do the math. Right now, we only have 4 this side, some type of papers for next semester. It's only one of the geography ones. So we just simply can't cut too many of them.
Yeah, so there's going to be feedback. Do you see a camera image?
I think we just need to turn.
It's just.
We can turn it just a bit like this.
Yeah.
Is that better? And Kathy's Kathy's like this, so
That'll be fine.
yeah. Okay, so you get the
Yeah, so that's fine. So we'll get that. That was it.
this is now starting to get hard.
Yeah, this is.
oh, it's because I see the camera is actually just a little bit. Oh. All right. Let me just. Okay, that's it. Yeah, there's just a little slider there. That should be okay now.
So that's a good run. Everybody. Yeah, that's much better.
Okay.
Got upset when we wanted to cancel ours, but
Kath, do you want to come just stand here for a second? And just, I guess, check the audio too.
Yep, so
Maybe.
I'll check over here. I think it's gonna get.
we'll get the reverberation, yeah? Okay. So the only thing is I won't know when you want me to talk, so should I do my usual and just keep talking, Martin? You know, I like to talk. So I was looking at pictures of Martin from 2015 and me, and I looked much younger and he didn't have any gray hair.
Shut up. And I
That's kinda
actually had some.
Oh, come on, your hair was awesome. So I was just like, wow, a lot has happened in the 7 years since we were
a
first in
long
the field. So, yeah,
It sounds
okay.
But the actual camera's not great, but I can't tell whether that's an internet issue. Like it's just.
Oh, the camera's not, yeah, it is. It's just
It's fine,
quality.
just to
It'll be
give
fine.
a good impression.
I couldn't find, there were some pictures I needed and they were no good pictures.
Well, that's great, Lucy.
So I went to
I'm happy then.
beautiful video.
Got it all set up.
Sweet.
I needed gas.
All good.
I can, letting us know.
Or it was really fuzzy or far away.
Yeah, it's not like it's just waiting.
So I'm just like, so finally level.
Yeah, I mean, I'll keep it.
This is a kind of practical and stuff.
Yeah, thank you.
And you can just send me the recording when it's done, if you like, bring it up.
Yeah, so Teams, the Teams meeting does that automatically.
Yeah, it's actually already recording, because I got a notification that you joined, like.
Oh.
But don't worry, you just keep it going.
Yeah. Now, how come I've got to?
And the whole thing then goes to astronomy.
No, I've had that before. That's okay. We can do that. We don't need that.
Still, you can find on this, I'm still on executive house. That's great.
Yeah. Okay. No, that's fine. Just so I can go in and manage it again if I need to. But otherwise, we should be okay. Oh yeah, so that is being recorded. Maybe I should just put that on hold for now.
I would just keep going, so we can trim as much of it as we like, but you don't.
Oh, can we?
Yeah, you don't want to start and stop it again. So,
Okay.
Yeah, you just.
Okay.
Email me the recording and stuff.
Great. Great. Thanks so much, Lucy. Ready to see you.
And it's very different. Not really for our students. You know, I bet you all do. Streamline it.
Now, is this totally set up?
Yeah, we're ready to go. We've got the screen share, cameras working, audio's checked. So you don't have to do anything except hit the down keys
Down
to
and
get
up.
it to move forward. Look, I'll show you. Down. Oh, yeah. No, we put it on display mode. Yeah, we put it on display mode. That's it. Yeah.
Now that thing, does that have to stay there the whole time? Because they're all gonna look at it.
No, we can get,
So if we can, and that way then they can see what's on the screen.
that's it.
Thank you.
So they've just got that now, and there will be a little corner view. But
Oh,
if you can stay here,
I can stay there.
and you can use the mouse
I'll
as a
use
pointer,
that. So I'll just
right?
stand here.
And
Yeah,
just
no,
do
that's
that
good for me.
relatively smoothly and stuff. So
That's great. All right,
that's all right.
okay.
Good. Right. So we're teaching this.
Yeah, I put 3 away from James. And this Jim. That'll be a lower impact. I was trying to get James to go over. I took it over for it, and it's basically a hot out.
She did.
Which I'm not a fan of now, because it's that whole,
So I was wondering if you buy in.
yes,
Now that's fine.
but it's trying to take it off to something else.
It is not okay. I'm gonna do it. So what's the point of being?
Yeah. Yeah.
Like, even just taking. I do want it to be the endpoint of the course. We ask the students for more science oriented. Now, I will have a whole other in it. And then what to do about the field trip. This. So, talking about hundreds of dollars. They have that. Right now, it doesn't work. So I'm going to make it like. You know how much work they are.
Yeah, both the days, which is why I think you should be working through. Yeah, of the 3 people on the teaching team, I'm like the lowest 3.
Well, in the high school course that we taught, JR and I was definitely a tiny one. And she brought in some of the I was the littlest part, and then she was always pushing me.
Yeah, I did we did more stuff for her 1 year.
So it's like, and I haven't asked you. We looked at for some of the good science teachers. Confusing because she's got another course that's similar.
No, I don't understand why she did that.
Never said anything. Teach it in my office. And I'm happy to have, I don't really have to go to the course, so I'm happy. Just piggyback. Stop doing
Well, it's like I wanted to revamp my illustrator course. It's not the student numbers we didn't really want to do. Yeah, I'm better off leaving it the way it is, because more than half my students are actually environmental science students. So if I, if I did it the way I wanted, I'd have 2 or 3 students. We just don't have the numbers. So I keep putting off my rebuild of that course. It's not that we're teaching that much, it's that the teaching for a course is double or triple the workload compared to what it used to be. But I mean, the idea of teaching a couple of classes is not that big, but once you start having to do all the additional stuff that they required of us now.
Yeah.
The workload per class is a lot more than, I mean, even since I started teaching, it's a lot more than it used to be.
The online
We've got a culture, and it seems to just be earth science, because geography doesn't do it, where we run the labs ourselves. And, in the States, you always have your TAs, and that's how geography is run. And I'm not quite sure why we have.
Like that's so right.
Yeah.
I mean, we could, but I feel like now everything's heading like a market set towards, that's when you really engage them. It's in
Oh,
those practices.
yeah.
Because of what's going on in not people starting to not do elections before, but
Yeah.
it's already going that way.
Oh, yeah. And I just didn't want to shut up. That's why it wasn't even online.
The university is pushing us to move away from your standard lecture format. They want the students to have a reason to come. It's not just asking to stand up.
Yeah.
It's like for my postgrad class, what I did this year was that we have a software program, H5, where the videos and have questions, pop-up questions that the students have to do multiple choice answers. So I actually built all of my lectures in that. So the students would have to work through multi-choice questions, and they get their points basically for doing it just by making it to the end.
Yeah.
But then it meant I wasn't doing lectures during the class time, and I just run, because it was intended to be a discussion-based class, and then just do the discussions during the scheduled class time.
We can do that with postgrad. I don't know about everything. Postgrad, I can see.
Yeah. I've thought about that for 203 as well, as a model for moving the lectures into the virtual space.
Yeah, it's so great.
Focusing the class time on the.
I know a friend of the city wanted to come, but he hasn't been in, so I'm not sure if he's going to make it in. He's like some extra report. Okay, yeah, I might just still be trying to help.
Yeah.
No, yeah.
So why not? Actually now,
This is how I'm unwinding for the day. Been on my feet the last few days. Chance to sit down. No, she's had a flu or something. So she's been like coughing for the night. So she was very keen to come in, which is like Yeah, post, and then she also went down towards Manchester one weekend. I was camping in the freezing cold.
Only 15%.
No, this is, some other people that come.
Once we were going,
Yeah.
so now the whole thing on starting over the whole thing. All of it is good, but
Yeah, it's great, yeah.
not super. Carol has one thing, so you're happy.
Yeah. So I can see when you see the. One was a huge I had three students out of the East, made up just the core postgrad class. And at postgrad, it's great on all they have to do is talk to discussion. Class and take part in discussion. That's it. And then I gave like three of them university. Just killed people. And I mean, yes, I mean, not a lot, because there's no like it's to do this. That's essentially what my discussion is on. It's already it's like that they show up as well. There was it's not
That's that kind of waste of your time to make sure.
Also, inside.
I don't know how to start.
Well, whenever you guys, I mean, we'll have to do that.
How they say that.
Yeah.
I mean, our first was.
Well, it's funny, because they're coming to the same thing.
Okay, now it's on.
And also there's a draw through things that are studied in this range. Our own distance sometimes. In these last 5 years, we actually hadn't seen it. And looking at it's called fraction of four. So it's quite
Reasonable number of free rate.
quite nice because again, you see these guys absolutely.
How many of those are science?
And then they're
4 out of 27. Maybe we struggle.
Yeah. Oh, it's amazing. And it's a snowball. All of that. It's really good. She was just, oh yeah, it's quite something that's, you know, one of these ones. I'm actually trying to get oh, it's really good. And then the main locality is now national park. It's a big fence, the guide comes from the inside the fence, you know, it's usually. It's a station on there. You right there, comes down. That's the institution. As far as that is, I can show you that first video. What do you think we found the oldest periods? The amounts of fine would be
Oh, yeah, that's. Okay, right, yeah. Yeah, once I'm all sorted, like, yeah, just get access to these as well, and that's fine.
That way we get all the. Well, if we do that, let's find out.
Okay. Hi, everyone. We're going to start very soon. So I've got a good intake on Teams. And you'll recognize some of the faces there. There was Carol right there. Okay. So yeah, you ready to roll? So I think, yeah, if you just name here, sorry. You might not enjoy it.
Don't roast me.
I'll try to be nice. Okay, everyone. I think we'll get started. Thank you very much for coming. I know it's an unusual time slot late on the Wednesday afternoon, but this is the time when the Australian Centre for Astrobiology has been regularly meeting for drinks down at the Roundhouse pub. Wednesday afternoons, we figure it's a good time to sort of be social. But sorry, it's a very good question. Yeah, we get that afterwards. That's right. So a bit about temperature, a bit to do with temperature. But it's my great pleasure to welcome Kathy Campbell to UNSW campus. She's been here a couple of years ago, we were saying, November 2019. So just as COVID was really getting to grips, but before lockdown, we had a workshop on a drill core that we obtained through the most convincing oldest evidence of life that Kathy won a grant in New Zealand for. And at the same time, I won a grant to study hot springs in New Zealand. So there's this cross-fertilization between deep time and the knowledge of active hot springs. So we've been collaborating now for 7 years or so. And Kathy's here as a visiting scientist, a fellow of the Faculty of Science at UNSW. And we are all going into the field on Sunday, back up to the Pilbara to look more at the habitats of ancient life and the relation of these 3.5 billion-year-old life forms to hot springs, which have been identified in that area. And so Kathy's been instrumental in us starting to recognize the extent of hot springs so very long ago, probably the oldest exposed land surface in the geological record. And when we first had her up, we were seeing a bunch of rocks, and Kathy says, I know what that is, I know what that is, I know what that is, and has made a huge contribution to our understanding of ancient life. And at the same time, we're using the chemistry and the fluid compositions and the complexity of hot springs to cast our mind to the origin of life, because hot springs are important for their variability, their potential to mix components of very different geochemistry and make complexity. And that's really the exciting thing that together with Albert Fehrenbach's group, looking at prebiotic chemistry and applying our knowledge from modern hot springs and deep time to really start to influence the origin of life field. So Kathy's originally from the US, but has been in New Zealand now for probably the same length of time I've been in Australia, 30 years or so, from the University of Auckland, trained as a postdoc at NASA Ames for a couple of years, and then got a position down at Auckland where she's been, forever and a day.
Yeah.
And very much like me, she's also a director of a center for fundamental inquiry to Ao Marama in Auckland that has a wide range of researchers addressing astrobiology as we try to do here. And we've worked together, but we're looking forward to sort of combining that expertise, because astrobiology is a very cross-disciplinary study with geology, biology, physics, all sorts of things, and nobody can be an expert in all of those. So our discipline is really trying to become a superorganism and applying our mental strength to that. But anyway, Kathy's here today to give us a seminar, as you can see from the title, on extreme environments of hot cold springs on Earth, and then spreading that out in the search for ET life, extraterrestrial life in the solar system. So thanks, Kathy. It's my great honor to have you here.
Thank you,
I
Martin.
look forward to your talk.
Thank you. Kia ora, everyone. Thank you so much for coming out this afternoon, and to those of you online today. I recognize a few faces of people not here who've beamed in. So it's wonderful to be here. I can't believe this time has gone by. We've been living in a time warp, and it's great for us, especially from New Zealand, to get out, because we have not been out and about, not even as much as you all have been. We've been really closed in. So it's my first trip overseas for a long time. And I thought I'd do something that I haven't actually done before, which is to talk about, in a way, about my career research, because I spent many, many years working on in marine environments. And it's only in the last, 25 years that I've started working in terrestrial environments, but with the same goals to use these extreme environments to help us understand very, very, very early life, as well as the possibility for life elsewhere. So this talk is divided quite into two bits. And the first bit is really about how we might apply marine systems, marine extreme environments. And in particular, my work was on cold methane seeps and gas hydrates, which are frozen methane, water, and ice, and the possibility for using those to look for life beyond the standard Goldilocks zone in the solar system. And so I thought I'd talk a little bit about that work and why and how geology kind of feeds into the context for our understanding of where to search, because it's a very big solar system out there. And even on Earth, there's a lot of rocks out there. How do you know where to look? What's your best chances of finding the best preserved life? And this life is very much dominated by microbes, as you can see from these two pictures. And then the other half, the second half, will be all about hot springs. We'll come into the inner solar system and think about early life on Earth, and particularly the application for searching for life on Mars. So it's kind of in two parts, and it spans my whole career in terms of looking at extreme environments, which, by the way, I stumbled completely into by accident as a master's student. And all I can say to the students who are watching or here in the audience is that the key was that I noticed something different that wasn't normal. And it bothered me, and it bothered me, and it bothered me to the point where, as a geology master's student, I went at the University of Washington to a zoology professor and asked if I could sit in on his zoology course to learn more about fossils, because I was having problems out in the field really understanding things that I was seeing. And one day, this guy put up a gutless clam from a hydrate ridge off of Oregon, and suddenly I realized that the whole world spinned in the lab. And I was just auditing the course. I was just a student. I wasn't even taking the course. But I think I was driven to find out what what was going on, having no idea that there was this thing called chemosynthesis or chemosymbiosis, or, you know, organisms that live without guts in the deep sea. I had no idea. And so I was very lucky that I was curious enough to try to figure out what to do. And went up to him afterwards and said, my God, I think I have one of the first fossil seeps in the world. And he said, here's the slides, call this person, do this, do that. And so I had a lot of great mentoring. So anyway, so I want to take you on a little walk through this to talk a bit about why we do this and how we do this. So I think our question today then will be, where should we look for life in our own solar system? And look a little bit about how studying extreme environments, or what we call Earth analog environments, might help us narrow that search for extraterrestrial life, past and present, and also give us some kind of context for all the fancy instrumentation that people use who go in and do, you know, really high resolution, for example, geochemical work. So my other message is context is everything. And being a geologist means we have to understand everything from the planetary scale right down to the microbe scale or the micron scale. So I want to try to illustrate some of that as well today. So just a little reminder, and this group probably doesn't need reminding that life has 3 basic requirements. We've got to find the stuff to make life, the organic building blocks, the energy source to assemble and continue life, and that source can be solar or it can be geo from the planet. And then the liquid medium, so used to transport the energy and transport the processes and assemble the stuff. And it seems that most worlds have the first 2, but there seems to have been a bottleneck in searching for life because liquid water in the solar system is rare, or maybe perhaps the question is it really that rare? And so let's just go on a bit of a walkabout to take a look at this question of habitability in our solar system. And we have this classic diagram here of the Goldilocks zone, which is the solar habitable zone. And through time, it's changed its position. Earth is at point 1 here, away from, this is the radius of the orbit. And then as the sun changes, the habitability changes through time. And that habitable zone is the too hot, too cold, or just right habitable zone. If you pick up definition of the habitable zone, it's standard to say, well, it's the zone around a star where liquid water is stable at the surface, is what they use. So we're going to come back and revisit this definition in a few minutes because, of course, it has definitely been expanded. And it's extreme environments that have expanded this definition. So the other thing I wanted to point out is in talking about the Earth's biospace, which is something that Charles Cockle has talked a lot about in his astrobiology course. So I thank him for this because I find it really compelling. And that is, if we look at life and conceptualize it with the tree of life, that's one way to look at it. Another way is let's take a look at its physical and chemical limits, or its extremes. So if you think of all of life on Earth as being inside a big zoo, and the fence is the set of physical and chemical limits. So beyond that, that's the difference between life and death. And then we can define those limits by several different ways. We can say, well, survival, one side versus the other, metabolic activity or growth or reproduction. And so we see that these extremes are all out on the edges. And these extremes can be of varying kinds: pressure, temperature, salt, water, and others. And the other thing about it is it's very important to look at the Earth's biospace and these limits, because we want to compare, to start with, if we find these kinds of conditions for life on other planets, and we know what the limits are, and they're usually extreme limits when it comes to microbes that are very well adapted to these extremes, then we want, of course, are always comparing to the only type of life we know, which is ours. There are limits to that as well. I'm aware of the fact that there might be life as we don't know it, but we often start with life as we know it. So we can expand that, the diagram showing that as we extend those limits of life, then the biospace gets bigger. And that's dominated the edges by the microbes. And that's just because as the conditions become more extreme, complex biochemistry of multicellular organisms gets more and more difficult to modify, notwithstanding the tardigrades, which are pretty spectacular and are multicellular organisms. But in general, single-celled microbes have an advantage here. And so they end up on the extremes. So we have these extremes. And so when we look at some of these physical, chemical limits of life on Earth, this is just a little list here of our current upper limit And this is all microbial, and of course these things are named. So up in the hydrothermal vents, up to 122 degrees Celsius, we have the hyperthermophiles, so the superheat-loving organisms, and then we have the cold lovers, the dry-loving, the salt-loving, the acid-loving, and the alkali-loving organisms. And of course, in New Zealand, we've got loads of hot springs, and we certainly are aware, this is an article in New Zealand Geographic about our understanding of life on Earth and our expectations of what life might look like elsewhere. So that was an article about extremes. So this is maybe a more accurate view of habitable zones with an S, multiple habitable zones in the solar system that shows both surface and deep habitats. And so here, this is from an article written about the JUICY Explorer missions, which is a European Space Agency mission that's going to launch in 2023 to go characterize Jupiter's system and look at some of these icy moons and really take a look at where liquid water could be stable at the surface and or in the subsurface, deep habitats of a celestial body. So that includes icy moons, not just planets. And as you can see, here the model goes beyond the snow line. So we didn't think liquid water would be out there, but of course it is. We see it in the moons of Jupiter and Saturn, and liquid water beneath the ice, and possibly even these methane hydrates that I was just mentioning at the beginning of the talk. So frozen methane and water, and life here on Earth being associated with that. So it's really a bigger picture, thanks to the work that's been done and the new missions that are coming out to be looking at these saltwater oceans and lake-like features underneath ice on several of these moons. The liquid is there because of tidal heating, because the two planets are just so huge. They exert such a huge influence on these icy moons. So they have ice tectonics, and meaning the ice is moving around on a body of something that's liquid underneath, and even geyser plumes erupting into space. So this is a very classic drawing of a cross-section of the crust of Enceladus with the ice moving around, the geysers coming out the southern part of Enceladus, which is quite a small moon, and this artist's conception of what's going on at the interface between the rocky core and the wet ocean, and the possibility of hydrothermal circulation or possibly cooler water circulation. And again, using models from extreme environments on Earth, looking at, in this case, they're considering the warm white smokers, but other groups in Europe are also looking at and considering whether there may be methane seeps and gas hydrates down underneath the ice in the oceans, down inside of these bodies. The other thing about them is that because of rock-water interactions, there's crustal fluid circulation interactions, minerals forming, salts and organic compounds have been detected in the plume in space. And there's also raining down from Jupiter or from Saturn, different materials that then could be used as nutrition for possible life in those oceans. It's dry in here. So the question is, do we have chemosynthetic communities similar to what we're finding in Earth systems down there in these celestial bodies? Now, this is also another aspect, is whether, if you look at the Tree of Life today, and there's a lot of things about this that are worth just mentioning. We've got our 3 domains here with animals way up here. The deepest roots of the living, most primitive organisms are hyperthermophilic, so that's the purple branches of the tree. Whether the last universal common ancestor was hyperthermophilic, as is shown in this drawing, is definitely debated, as is the question of whether or not there has been horizontal gene transfer between archaea and bacteria, so that making this look like life originated in hot water. Those are all being questioned, but there's certainly no doubt that hyperthermophiles sit down at the base of the Tree of Life, and perhaps these heat-loving microbes are a remnant of our impact bombardment phase in the solar system's early history. So here we see some of these microbes, the pink microbes in a hot spring on land. That's this, not this exact one, but this branch. And then we have, of course, in the black smokers, some of the most heat-loving of all microbes. And so the chemosynthetic food web then was discovered in the 1970s, well, into the early 1980s, first in the hydrothermal vents. We have both internal symbionts or chemosymbionts that are living in the cells of organisms like the tube worms or clams that actually utilize the geofluids of the earth with no light, and they then release energy and build up organic molecules, so these organisms don't have guts. There are also microbial mats that live on the surface that also utilize the hydrogen sulfide coming out of these vents. In this case, these are cold seeps on the right and black smokers on the left. And so this extension of the edge of the biospace happened in the 70s and 80s in terms of our understanding of what life could live, how life could live. Seeps in particular are often found in very active plate tectonic boundaries where you have subduction, convergence, and organic matter coming out of the seafloor. It's been processed by microorganisms, and methane has been generated and then oxidized. And there's a whole food web here. And of course, in the hydrothermal vents, these are divergent plate boundaries with hot water coming out. That was predicted by geologists. So the very first cruise didn't have any microbiologists or biologists on it. It was only geologists who found the springs. A bit of a shock when they found the life associated with it. Now, in terms of seeps and in terms of my own work, what happens, we now know, is that methane is in seep provinces is coming up in relatively high concentrations, often in subduction margins where the organic material is being squeezed as the plate subducts, and we get leaking oil and gas all over the place. And in that sediment column, with all this methane and hydrogen sulfide being generated in the sediments, we have an excess of alkalinity that gets generated when anaerobic oxidation of methane is happening with archaea, and they are in association anaerobically with sulfate-reducing bacteria that create the sulfide that's then utilized by other microorganisms. And so you can end up in a seep province with gas hydrate, which is frozen methane, water, and ice that's stable at the sea surface, and carbonate accumulates. And carbonate is what gives us our deep time rock archive of microbial biogeochemistry. So it's in that rock archive that we can then work and look at the history of the seepage and look at biogeochemical cycles in these rocks. So that's a much more permanent archive than even the gas hydrates. So in the 2000s, I was working, I started out working in the Western US. As you can tell from my accent, I studied over in the Western US, and actually at NASA Ames, I was asked to come and look at microbes and their preservation in extreme environments in hydrocarbon seeps. They were all working on hot springs, and I was invited because they were very generous. They wanted to see if there was an interesting story in the microbes, and they understood that this was another kind of extreme environment before we knew about Enceladus or Europa. So around the Pacific Rim, we can see some important things. That's where I've been mainly working, in New Zealand, Japan, and Alaska. In any case, we have this record of the divergent plate boundaries and the hydrothermal vents along the divergent plate boundaries shown here in red. The modern seeps there are many more that I'm showing, but the well-known ones with a lot of organisms associated with them are shown in these blue squares. And then the fossil record of some of the more important ones are shown, the vents in yellow and the seeps in green. And there is some species crossover between the two in the extreme environments. You know, you get giant clams and tube worms and both, and lots of microbes. And so you have, particularly around the Pacific Rim, which is so tectonically active, there are really a lot of seeps and vents. And you can also see that the geological record is giving you hints of past plate tectonic activity. We're here in the middle of the Ural Mountains. You'd be wondering, why are there hydrothermal vents there? And that, of course, they're 400 million years old. Massive sulfide deposits, but ancient ones. So plate tectonics and evolution have created vent and seep provinces. There's been exchange of organisms between them, and we're interested in, you know, when did that first happen? What kind of a planet do you need to make this happen? And the answer is you need plate tectonics for sure to have this kind of dynamic thing going on. Or maybe ice tectonics is also possible, and possibly that tidal squeezing on some of the outer icy celestial bodies. But you need some kind of dynamism to get the water-rock interactions going, and preserving, making minerals that preserve the microbial cycles. So I won't go into all the details of all the work we've done, but I wanted to just point out a few things about the seeps and then kind of have a take-home message about them, and then I'll go on to talk about hot springs and Mars. But in terms of seeps, important things to note are that you can go out in the field, and when you're working in rocks, the nice thing is that you don't have to have a submersible. You can walk around. This was when I first moved to New Zealand. These trees are now gigantic, and it's very difficult to work here now. But at the time when I first arrived, we went out to look at the largest paleo seep. It's about 20 million years old, out in the east coast of the North Island, which is right where our subduction margin was getting going at this time. And you can map out distribution of the carbonates, and that's what this is here. This is an oblique view. And this D outcrop is this one right here. And you can map out vertically and laterally the distribution of the organisms and then study the carbonates. And so some of the things we found is that the organism distribution is very much tied to the fluid flow rates, relative rates. And you can actually look underneath the seafloor. So the seafloor line is drawn with this dotted pattern, but underneath is all the subterranean fluid plumbing that was coming up. And there you have conduits that are actually bringing the fluids up from depth and then up onto the seafloor. So there's a bunch of information you can get out of doing this kind of mapping. And some of the conduits are gigantic. This is the biggest one that I know of in New Zealand. These ones are donut shaped. Maori used to use these things as anchor stones when they were fishing out of their canoes. And so they actually have cultural significance, and they're shaped like this naturally. So these are subsurface plumbing features, but you can find places where the tube worm thickets are full of particular types of cements that tell us that the fluid flow was very vigorous and very advective, and you get a very strong AOM, or anaerobic oxidation of methane signature, out of these. Where the mussels are, it's more muddy, it's further out, and so these zonation patterns. And further out on the flanks of the larger features, we have broken pieces of rock, but diffuse fluid flow. You get a whole different suite of organisms, these clams living there. And so you can actually reconstruct the history of the fluid flow, and you can do that over the whole margin and around, let's say, the whole Pacific Rim over hundreds of millions of years using this kind of mapping style where you look at organism-sediment relationships and reconstruct the fluid flow and how much fluid comes out of a margin, a convergent margin over time. With lipid biomarker work with our German colleagues, you can actually then find biomarkers for AOM that go back at least through to the Devonian. These ones go back here. These are microbialites, thrombolites. So these are a type of, they're not stromatolites, they're the other version, which are the more clotted microbial features. And so here they're encased in this fibrous aragonite that is in the peak of the venting. And so you can really learn a lot about the distribution of organisms and what the microbes are doing over time. And if you couple that with, I did a big review a few years ago, trying to understand why one of our big data sets that everybody uses in the SEEP world, which is stable carbon and oxygen isotopes, why that's so variable, and is there any pattern to any of this? So I looked at hundreds of papers, modern and ancient. It took a long time. Review papers are a lot of work. But what I found was that if you look at all the data, what you can do is see that there's an isotopic fractionation of the oxygen and the carbon isotopes that help you understand the source of the carbon and the processes that are undergoing in the methane seepage, especially. You get this very strong fractionation and very negative carbon signatures, what we call sort of classic seep carbonates. And all the young, almost all the young deposits show this feature. And then you get seawater at 0 and 0, and all the shells of these organisms are basically being formed biomineralization-wise by seawater. And they're living in seeps that are bathed in this isotopically very distinctive carbon signature of the dissolved carbonate, but the shells are not picking this up. And if you're in a gas hydrate province, then it moves the the oxygen isotopes up toward the positive, which is much colder. So if they dissociate, then you're basically melting the ice, and the methane source is coming from this microbial component that has a very positive signature. But when you bury these rocks, burial diagenesis is also warm, and the fluids have a different composition. And so the old rocks are all being pulled because they undergo diagenesis, and this affects your primary signatures. The other thing that we found, there's a bit of a mineral isotopic fractionation, that's dolomite, high mag calcite, aragonite, and calcite, and it has a small effect on the oxygen isotopes. The other thing that I found is there's a group of modern seeps that are very weird, and they have very weird isotopic signatures, as you can see. But if you look at their tectonic settings, they are in places where there has been deep sea erosion. And what's happening is that they're tapping deep fluids that are coming out on the seafloor. So a good example of that would be submarine canyons, like Monterey Canyon in California. There's a deep submarine canyon right there at the coast. It's a subduction transpressive margin, and there's fluids that are coming out that just are not normal. They're deep burial fluids. And so what was really nice about this work is we began to make some sense of all these carbonates and all these signatures. I had somebody write to me from somewhere in the world, and they could not believe some of these signatures. So I just sent them all the original papers and said, well, here you go. Here's our idea of what it all means, and let me know if you have a different idea. So that was nice to be able to put the work I was doing, because I was working in this space right here, and try to understand what the signatures were telling us in terms of the biogeochemical processes. Now, the cycling at methane seeps is pretty complicated. Organic matter is coming in. It's going through its normal decay in the sediments, where you're producing a lot of methane by some microbes. Other microbes are eating it and keeping us from going into a full-blown global warming, because this happens all over the seafloor. In seep provinces, there's so much methane coming out that you get bubbles on the seafloor. And of course, they're worried that all the gas hydrates are going to melt, and then we'll end up with this big spike of methane. So there are lots of different steps and different parts of the processes that the Germans have mainly been working on that contribute to the isotopic signatures that I just wanted to point out. I don't want to go over it in any detail. My work was really to then try to put the fossil record and the record of seepage into context and understand what this complexity in all these carbonate cements was telling us. Complexity is a signal that interesting things are going on with the fluid microbe mineral relationships. And so this is a Devonian seep rock. This is bitumen, the black. These are big brachiopods. The light gray is like marine sediment, that's diffuse seepage, and the very light colored fibrous cements represent much more vigorous seepage that you can look at in terms of elemental composition and stable isotopes. And I won't go into all the details here, but by doing a combined, this is my postdoc at NASA, by looking combined at the stable isotopes, the elemental geochemistry, and putting them in a framework of what comes first, what comes second, what comes third, which is called paragenesis. It's the history. Then you can actually reconstruct how fluids change through time, how the organisms are affected by that, and what the characteristics are of your extreme environment. This is a Cretaceous example. These are tube worms. Here's some big tube worms coming across. And there's a huge amount of information in this, but you have to make sense of it in order to understand this complexity. For example, in this Devonian example, the bitumen is one of the latest features. So this is fossil oil, and that fossil oil migrated into the seep at the very end, for example. So just taking one example of some of the work I was doing at NASA, this is a tube worm. So imagine a tube, the animal dies, it's being cemented by the seepage because carbonate's coming out, and there are different phases of cementation, and as it occludes with the cement, you get different types of phases that are telling you about what's going on in the history of that seep. And so I'm only selecting a very small thing, but basically if we zoom in on this tube worm here, we see a lot of different, these clotted fabrics. So there's two microbial phases, M1 and M2. M1 is very late in the piece, right before the seep deposit is buried and fully occluded with later these are burial cements that come after, so it's like a puzzle. You figure out what was first on the seafloor, and then when this organism dies, these cements fill it in, and as you do the elemental compositions and the isotopic story through time, which is from one to the next one to the next one as it fills, then you can actually reconstruct the composition of those fluids and the conditions under which the microbes were living. So this is the point of my postdoc. And so we see that at the end of the early diagenetic seafloor phase, we have microbes that were living on the seafloor, as well as ones that were living inside the cavities of those tube worms. And they have now shown in modern seeps, long after I did this work, that indeed a modern seep carbonate chimney is full of microbes, even when it's solid rock, they're still living in there. So this is the story of what happens first on the seafloor and then what happens during the burial conditions at different sites. And then you can reconstruct these relationships. So these are the kinds of tools that I would want to use if you're bringing back materials from places where you are have an under ice sea and you have deposits that are accumulating. I would be looking for signatures of microbes and minerals in association with one another. So it's going to take a lot because some of the ice in those icy moons is something like, 70 kilometers thick. There are places where the ice tectonics is thin, and they're talking about they have instruments that are going to drill down through the ice one day and go into submarine, like little submarines, and fly around and try to find these vents. So that's work that I think has been extended by studying extreme environments on Earth. So that was early work I did, and it got me to NASA. And when I was at NASA working on these seep microbial signatures, I was taken out to Yellowstone because the whole team worked at Yellowstone. And I learned about these extreme environments at Yellowstone and their relevance for searching for early life, particularly the question about whether they might be relevant for searching for life on Mars. And here we again have microbial communities living around hot springs, and where the water discharges, you see all the different colored mats. This is the Grand Prismatic Spring. It's not at high boiling temperatures. It's a little bit lower than that, so the cyanobacteria in particular thrive there. So you see the oranges and the yellows and the browns. Those are all cyanobacteria that are living under optimal conditions for them. And where you see the white are the minerals that are coming out of the springs and entombing them and preserving them in vivo. So here in New Zealand, where I work, we are looking at microbial communities and how they turn to stone. This is a modern hot pool with all these microbial mats in the summertime. They all have carotenoid pigments, which tell us that they are dealing with sunscreen and UV radiation. But when you go into the deep pools, all the stromatolites turn quite bright green. And so these things are being silicified. All the white that you see are the silica that is actually infused in hot spring waters and is entombing these microbes. This is a 60-year-old hot spring deposit, and you can still see it has carotenoid pigments. And you can see that it was killed off by some kind of a storm event, bringing in detrital rock pieces. And so this rock material, this silicious, silica-rich hot spring deposit is known as sinter. So that's what we work a lot on, and I work with Martin on hopefully we're doing more work, in a week or so, but we're trying to understand more about these ancient hot springs in very, very old rocks in the Pilbara. So what happens is the mats dry out in the summertime or where there's no flow, and they turn pure white because, of course, it's silica, and then they're entombed with all their textures that tell you about environments, reflecting very specific hot spring environments, whether you're in pools or you're in the vent itself, which is over here, for example, or you're in the discharge channel. They all have different shapes, different textures, and we call those on Mars, when you see them, potential biosignatures, the actual textural material that helps us hone in on where to search for possible life, and in our case, for identifying where to sample for a Martian sample return mission. Regards to early Earth and early Mars, the link is that 3.5 billion years ago, both planets were anoxic, both planets had volcanoes, both planets were experiencing impacts, bringing heat also to the planet, and both, of course, had liquid surface water. So on Earth, we had hot springs where there's life, and so the question is, what about on Mars? And so luckily for us, Spirit rover was there from 2000, well, it's still there, but it was active 2004 to 2011. And here it was at home plate, which is a volcanic ash deposit, 80 meters in width. Here's a photo that was taken from orbit of Spirit. It ended up dying over here later on. And the dashed lines show this area around home plate that is full of finger-like or digitate silica features that you can see here. And so this was discovered in around 2008 was when it was published, around 2007. And when they did the thermal emission spectra of the home plate silica, which is shown here in the black, they found that sinter, silicious hot spring deposits from Yellowstone or from El Tateo in Chile, have really strong matching for these thermal emission spectra. So it's opal A on Mars. On Earth, rocks this old would all be turned to quartz by now. The silica, the opal, the non-crystalline silica turns to quartz over time. But here on Mars, probably because of not very much water, no plate tectonics, and not a lot of, weathering or turning things into going through geological cycles, it's still opal A, plus salt. And so on Earth, these hot springs that are salty are the ones that come from alkali chloride waters that interact more deeply with the magma chamber that's heating up the circulating meteoric waters. And so the match is actually closest with El Tatio, which is at high elevation, low water flow, thin atmosphere, and nothing washes away, so the salt is still there. So the match looks like we're looking at hot spring center on Mars. Now, I'll come back to that in a few minutes. I just wanted to say a few things about hot spring systems. There's a whole range of fluid chemistries, and the question is, when they first were talking about going back to get samples from Mars in the Mars 2020 mission, Columbia Hills was on the map of being a possible mission, and NASA was very, very worried that the silica at Columbia Hills was this kind over here. This is from Hell's Gate, Tikateri in New Zealand. This is an acid system with low pH acid sulfate, mainly destructional with lots of corrosion. Silica is there, and there's a little bit of silica sinter that forms, but it's mainly, most of the sinter is remobilized and released from the acid. And there's a lot of other minerals like sulfur and iron oxides and other minerals. Whereas alkali chloride sinters are near neutral to high pH. They build up big sinter aprons, so big, huge areas of sinter buildup in association with the microbes. So the microbes are embedded in it. There are microbes here for sure, but they're more like in biofilm. And so we have this pure silica composition that is really, really good for preserving microbes. Like we've spent a lot of time, growing the microbes with the silica, growing the stromatolites. There's all different kinds of stromatolites, and seeing how they grow and how they preserve. And they do very, very well indeed. But NASA was super worried that the silica on Mars was actually of this type. And they thought, well, this would just destroy whatever was there, and there's nothing left. And so why should we go there and spend a total of $7 billion in the end to bring samples back if there's going to be nothing there? So in brief, if you have a magma chamber, and this is just where all the fluids come from, it's meteoric water that's coming down and circulating and being heated up by the heat of the magma in these shallow hydrothermal systems. That water rises, it interacts just like what they're talking about on Enceladus, water-rock interactions, collecting elements, and it gets to a point where it goes through boiling at depth, where the pressure is released enough that you get boiling, and where the boiling separates out steam, the steam is acidic and it rises, and that's this red part, and you get very acidic steaming ground, and you get lots of hydrogen sulfide that's carried in the steam, fumaroles, mud pots, acid springs, and pits. So this is what NASA was worried about finding on Mars. They worried that there wasn't going to be much life here. If the water goes straight up and boils but continues to flow, the chloride fluids coming from at depth, then you can get thick sinters forming, alkali chloride springs, constructional sinter, geysers, and luxuriant microbial mats. So this is the fun fight that was going on in 2017 and 2018 when the decision was made around whether to go back to Columbia Hills or go to one of the other two locations. And you know the outcome of that. So I don't have to go into that too much. But we still very much hold out hope and planning for a Mars sample return mission from Columbia Hills. And there's reasons for that. And the reasons for that I want to show you in terms of looking at these systems. So we spent a lot of time looking at the gradient in alkali chloride systems from the proximal apron, where all the vents are, where the geysers are, all the way out to the marsh. So we've got a temperature gradient here. And as you can see, the mats in general are very, very thick. You only get biofilm up here because these are not cyanobacteria like all these pictures. And you can see all the different textures. It's spectacular and tells you all about the different environments, whether you're in a channel, whether you're in a shallow pool, a deep pool, and everything is infused with silica. Everything initially is very well preserved, all microbial and all preserved it's like the best environment to find extreme life. And so the digitate features are shown here, a couple of places where you find them, but there are all kinds of other features as well from the vent right out to the marsh that are all microbially controlled. And for example, our work in the Jurassic of Patagonia, which back in the day was the opening of the South Atlantic Ocean. So it was a rift basin, just like in New Zealand, and there was hot springs all over the place and lots of volcanism. And you can go out and find 150 million-year-old vents with vent geyserite. And that geyserite clearly has, if you look at it on a ramen, clearly has biofilm in it, which is the green, inside the quartz, which is the yellow. It's the silica's turned to quartz. And so there's no doubt that geyserite, which for a long time was thought to be abiogenic, because it doesn't look like it has anything in it, is full of life. And so what you're looking at is the highest temperature life on land anywhere on Earth is in the vent area, and the type of center is known as geyserite because, of course, it forms in the splashing of geysers. And so these are types of stromatolites, microbial biofilms. And so we now know that the whole hot spring system is basically a microbial factory of all different kinds of microbial life adapted to all kinds of different settings. So that is a huge tick in favor of going back to Mars and sampling what looked to be silica-rich hot spring deposits. And in fact, here you are. Here's a hot spring, alkali chloride hot spring deposit with the digitate center here in New Zealand. And here is a close-up from the spirit of this digitate silica on Mars. And, there are people saying, oh, this is formed by wind-blown features, or maybe they're corroded and eroded. And so we're in the middle of doing more work on this, but there's been quite a bit of work done to show that most likely these are actually primary features. But we don't know if they're layered and laminated. We don't know if they have microbes in them. And you're not going to be able to figure that out on Mars. So the digitate center, where does it form? It forms in the outflow channels and around the rims of pools. And it's found in both alkali chloride environments, like here at El Tatio on the left, where the digitate features are a bit more knobby, or it's found also in acid outflow channels, like here at Hell's Gate, where it's a bit more thin and needle-like. But it's very, very common across different hot spring environments. This is one of our students who's now working with Martin and me and Michael and others on the dresser core, but she did her honors work here at Hell's Gate. This is a walkway, this is people. And here at Hell's Gate, this is an acid system. And you can see the spicules, or the thin, narrow center, is found around the rims of pools. The blue things are pools, and here are the outflow channels. And this picture here shows the same pools and the outflow channels, and look at their different chemistries. And look at that mixture. I think this pool, Mike, the black is pyrite, not arsenic, right? Yes, so this is a iron sulfide rich pool. This pool obviously is more native sulfur feeding into this acid lake. And you can see the silica-rich digitate features are found in the thermal outflow channels and streams. And so this is the kind of thing I would want to map on Mars and then go out and sample. And that's what I would want to do. And speaking of Mars, here we have an oblique view of Columbia Hills, Husband Hill. The Spirit rover came through Husband Hill, down, around, and then explored Home Plate. And what you can see is there is plenty of sulfur-rich soil around, which would indicate an acid environment. Sulfur-rich soils, acid. So there was fumarolic activity on Mars to create these acid soils. And silica, especially around Home Plate. The problem in 2008 was they couldn't reconcile these two, but we've done a lot more work since then with our colleague Steve Ruff over at Arizona State. And we see quite clearly that you certainly can have both because of water table fluctuations. You can have the water table drop and you stop depositing silica center. And instead you get the acid steam and you can get sulfur-rich soils as part of the evolution of a hot spring system. So we can get both, and they indicate hot spring activity and do not preclude there being life in these digitate features. And just to double check that, to make sure that we were on the right track, we had a PhD microbiology student, Kitty Sriaporn, who looked at digitate center in New Zealand hot springs at all the different chemistries. So acid sulfate chloride, which is a mix between the two. There are some bicarbonate springs that have more bicarbonate in them, so we have some of those, and the alkali chloride. And you can see in every case, they're all forming right at the air-water interface. So they're not forming underwater. They're not domal stromatolites forming in a subaqueous environment. They're forming at the air-water interface by capillary wicking or evaporative wicking, and the microbial mats are inside. These are layers showing you the lamination and some of the different microbes that are in these digitate features. So the microbes are in all of them. Doesn't matter whether it's alkali, chloride, acid, sulfate, or a mixture. Life is in all of it. And so there is no problem with going to Mars and taking a look. And so I won't go into this in detail, but if you just look at the pattern of her work, this is Kitty, just finished her PhD. We have different spring sites in New Zealand that all have digitate center. And here you see the water chemistry on the left is more acidic, where it's red, and these sites are more alkaline over here. And the temperature, they all have varying temperature ranges. And you see the pH being acidic and alkali, and 8 pH, so very alkaline. And what's really interesting is that the microbes are found in all the chemistries. And what changes is the microbial community composition because of whether you've got acid springs or whether you have alkali chloride springs. And so, for example, this yellowy orange is cyanobacteria. And what do we see but thick cyanobacterial mats in alkali chloride springs? So she's really confirmed quite nicely that the digitate center is full of life. It's different life depending on whether it's acid or alkali, but all of it creates the digitate feature. And the other thing, some of her work has also shown that some of these organisms are adapted to high temperatures because they have genome streamlining, so their genomes are more simplified, and they have some thermostable aspects to them. So that's some recent work that was published just last year. So we're very confident that these digitate features are, in fact, microbial stromatolites. And so Michael Rao, who's here because he's going in the field with us from Auckland as well, our igneous geochemist and hydrothermal geochemist, we're co-supervising a student here for her honors, Emma Nursasova, and she's been doing CT, micro-CT scanning, 3D imaging, and looking at density changes of lamination of these digitate centers in New Zealand and also El Tatio, and trying to understand how easy or hard would it be for a rover to go to Columbia Hills and pick up this center and stick it in a bag and put it on a Mars ascent vehicle and bring it back to Earth. So we're actually working on that. We've got a big project with here with people in Australia, Martin, in the US, in Japan, and our team in New Zealand. So looking at that from a material properties point of view. So I think I will then mention just briefly our work here on early life and hot water on land with the Dresser Formation and Martin's student Tara Jokic, looking at, she went out and mapped across in the Pilbara and the Dresser Formation, so 3.5 billion-year-old volcanic caldera with what looks to be an emergent landmass, a lot of hydrothermal circulation and evidence for that. And she went out to find, because the plumbing was everywhere, the veins that carried the fluids were everywhere, but she went out to map and find the surface manifestations of what should be a geothermal system out there, and found this material here, which is pretty much a spit-in image for geyserite here in New Zealand. So this is the Dresser interpreted geyserite, which is really incredibly similar and matching what we see geochemically in centers in other parts of the world. And in the core that we drilled, that Martin mentioned, was drilled clean with water, no oil and other nasty things. This material was found by one of Martin's postdocs, and we've been working on this fabric. Michael was just looking at this on the synchrotron last week in Melbourne. I'm trying to see if, do we have something like this, which is a microbial center fabric found in Patagonia, and as you can see from the ramen, definitely organic. And the problem with the Dresser material is that it is basically clean quartz and has no organics in it. So we are working very hard to figure out, is this pseudo center or is it really center? And the jury's still out on that problem. We're still working on that right now. But that's coming out of the core, some of the nice material that we found in the core. A brief shout out to the Origin of Life, the advocates for the sea to land transition, hydrothermal vents or clays or all the other different hypotheses for where life came from on Earth versus the land to sea version, and the advantages of hot springs as a possible origin, terrestrial hot springs as a possible origin of life for both the mixing of the different chemicals and the different types of pools, as well as the wetting and drying cycles to hopefully create protocells, is the baseline for what would have perhaps been optimal conditions for the origin of life. And back then, maybe the oceans were the extreme conditions back then. So I won't go into that, but that work is being done by quite a lot of people, including some people here. So to wrap up then, this is a really beautiful scanning electron micrograph of one little digitate center forming at Champagne Pool in New Zealand, which is probably the most famous hot spring in New Zealand. And I wanted to point out not only the microbes that are being silicified with tiny little balls of silica, but they exude this gooey stuff. If you've ever touched a microbial mat, it's gooey. And it's called exopolymeric substance, or EPS. And the microbes exude the stuff to try to maintain the environment in the optimal conditions. And then they grow another layer of filaments. And the EPS itself becomes silicified. And it makes the lamination that you see in the fossil record, where you see microbes, and then silica, microbe, silica, microbe, silica. It's all biology, because the EPS itself preserves better in some cases than the actual filaments. So we certainly see that out in the dresser. There's lots of this EPS type stuff out there as well. So stromatolites then are important in the early Earth environments, hydrothermal systems, and in the search for past life on Mars. And we certainly have these digitate stromatolites that are morphologically similar to the home plate digitate silica, but we don't know yet if that silica is laminated, and we don't know yet, of course, if it has fossils in it. So we need sample return in order to determine where we all think is one of the best places on Mars. To narrow that search, instead of looking for a needle in a haystack, let's go back to Columbia Hills and get these samples, sample this digitate material, and get it back to Earth and do the work that needs to be done to find out if there was life 2.0 in the solar system. So on that note, I will finish. And I want to say thank you very much for your attention in the afternoon on Wednesday. And it's great to be here and out of lockdown and back and going to the Pilbara. So thanks very much. Take any questions.
Right. Cassie?
And that is Champagne Pool, named because of the carbon dioxide that bubbles 24/7 out of it. It's quite spectacular.
Beautiful. So listen, we've gone pretty much right to time. So if anybody has to go, it's 5 o'clock, please feel free to do so. But if there are any questions, we're happy to stay and have a chat, and also for people online. So let me just see if there's online for the listeners. If you have a question, please just raise your hand virtually and we'll get to you. Is there any questions from the audience? Okay. John?
Hi, thanks, Kathleen. I'm relatively new, curious about the origins of bacteria in the Arctic. That's probably right because they should be common, but I'm wondering about your distribution of species, particularly. Is there any clue to environmental selection that might have taken one style
Good question. It's a microbiology question. What I do know is that up in the vents, what we have are archaea for sure. So you lose out the bacteria largely in the vent environment in the terrestrial hot springs, and you have only the archaea. I do know that people talk a lot about horizontal gene transfer as why the bacteria picked up the hyperthermophilic sort of lifestyle. And I know that even today, and Earth is today's Earth is a hard, it's not obviously the best analog for these things because we have oxygen and we have a very complex life, but we see in some of the hot springs in New Zealand that as springs go up and down monthly, and if they have different chemistries, then students of mine who've worked in microbiology have actually documented the swap over in the dominance of the proportion of archaea versus bacteria on a monthly cycle when these springs go up and down, and the heat always goes up when the water level goes up. And so archaea then become dominant when the water levels are high, and they come out and they go down into the thermal streams and kill all the cyanobacteria that are just living there. And then again, there's a swap in the temperature When it gets lower, then the bacteria begin to dominate. So whatever is going on, at least in terms of the hyperthermophiles, the archaea seem to have the upper hand. Now, whether the base of the tree of life looks like it's all hyperthermophilic because of horizontal gene transfer, that's what quite a lot of people are arguing now.
The horizontal gene transfer in bacteria. At the end of the bacteria,
Microbiologists, let me know.
Next question.
Yeah, I don't know. Yeah.
Why do they take from a protocell that then adapted to two different sorts of early, one spring, one something else?
Want something else.
Or specialize at some point in this fluctuating cycle?
I think we have, there's a limit, I think, to the record. And the reason I say that, we may have to find the answer in the living part of the record or in prebiotic chemistry experiments. And the reason I say that is that bacteria, generally speaking, preserve better than archaea. I mean, in terms of us looking for them. Yes. And so in geology, we call it taphonomy, which is everything that happens to an organism after it dies and before it becomes a rock. And taphonomic processes are very bad for archaea. So we have this problem of not seeing them because of preservation. Histones,
Like we do. Which bacteria don't really do that? Eukaryotic cells have an archaeal foundation.
That's one of the, so one of the connections with archaea and eukaryotes. Yeah. Yep.
So that's curious too, because just those are there to stabilize. I guess that's
it's still open.
Yeah, because the two types of bacteria and archaea, the two types of cells are quite different. They've got different outer membranes, different internal structure. They are certainly quite.
Something in the early environment that's like for that specialization.
Yeah.
Structure.
Yeah. And I don't know that question. Is there a question in the back? Sorry? No?
All right. Well, cool. Thank you. Jeremy, yes.
When
So the question is about the preservation of bacteria, in particular bacteria with a lot, cyanobacteria, which of course are more advanced. So I'm sort of don't know what the earliest bacteria were like, but they were certainly anoxygenic and all of that. But the dominant groups that are in the alkali chloride springs in the sort of mid-temp, rather than up in the vent, but the mid-temperature, all have these very durable sheaths that silicify really well. And that's why all those pictures, they looked fantastic. You could really tell it was a microbe. And it's because they're sheathed, and the sheaths are of a particular material that just infuses with the silica like instantly. Like while they're alive, they're busy gliding out of the silica, moving upwards and trying to get away from being entombed permanently. So it's just a natural preservation quality of those bacteria. They preserve very well.
And there have been studies, there are papers about people trying to silicify microbes, like through active experiments. And they found that, the archaea have very thin cell walls, so they get actually replaced by silica and wiped out in a matter of 15 days or so.
Yeah.
Whereas bacteria, because of the different composition of the external part of their cell, it's more resistant and it doesn't allow the silica in.
Yeah.
So they can be preserved for much longer periods. And that's what Kathy suggested in the rock record. The preservation of archaea, partly because they're also very small. It's very hard to see. But it's also because the chemistry of their system doesn't help them preserve very well.
Sorry, is that any difference between chemically between that deposit and bacterial?
Nobody's really looked at that side of it from the actual geochemistry part. Like if you have a massive archaea, does that have a distinct geochemistry from a massive bacteria?
Full structure.
Well-
Structurally, texturally, yes, to some degree.
So you'd be looking at both of them.
You can, certainly the dense geyserite is much more finely laminated, and that is definitely more archaeal dominated or super high temperature bacteria. So the problem we have is that if the organic matter goes, which it does, and then all you have left is those macro textures that all of you could see very easily, we can see it, and you walk up to this thing and you say, that's a stromatolite on Mars or an early Earth, then lots of other people will say, well, that's not, it could be a pseudo-fossil, because we don't have the organics, so therefore you're back to zero again. So we have this problem of, that's why I emphasize the context so much, the environmental context of the organisms, their distributions, because you need to understand the context, not just some random sample that you're saying, is this life or is this not life? It's a whole system that they are adapted to living in. And as I said, in the seeps, they are actually adapted right into the rock deposit until it becomes buried. So we know where they are and where they survive. And you need that in order to counter the people who say that thing is not a stromatolite. Because there's lots of people who say, because we have these other things that form inorganically that look a little bit like stromatolites, it means that all stromatolites then are in question. And that is a huge debate and argument that we have ongoingly in the community.
These things are
The carbon goes.
It very easily remineralizes to either CO2 or to carbonate minerals. It actually remineralizes. And so, yeah.
That's organic.
Well, you can just have gases pass through the system, or
But you can have inorganic carbonate. So if you look at the Martian meteorite ALH 84001, which was a big news in 1996, it was an igneous rock that had carbonate globules in it with rims of magnetite. And people were saying, well, magnetite and carbonate are formed by the byproduct, mineral byproduct of bacteria. And other people said, but this can form by vapor. It can form inorganically. And so we come back to the same debate of what is a real biosignature. And so that is why I feel like you have to work with the system as a whole, not just say, okay, I'm going to go grab one of these samples and run, but really understand the chemistry of the system and the preservation state of your fossils. And so we spend a lot of time doing that on rocks on Earth, some of which you can get very young rocks that are completely munted, or you can get very, very old rocks that are in perfect condition. You know, what's controlling that? That's the environmental conditions and the depositional and post-depositional conditions. You've got to put them all together to understand your fossils.
So currently, the preferred sort of biological identifier, biosignature, is a complex organic molecule that can only be made by life.
Yes.
But just like DNA and dinosaurs, they have a very short residence time. They break down to simpler molecules that could be biological or abiological. So if you only find relatively simple organic molecules without a context in which to place it,
like on Mars,
actually don't know whether it could be life or not life. And so for us, we're context people, textures, all of those things have to be integrated together, which is what Kathy's really trying to emphasize.
Yeah.
Jeremy?
Silicification, right.
Just period.
Right.
They are. They are. The products of their metabolism are different. That extra polymeric substance is a different style. And so, yeah, the preservation is different between the two. And remember, we're never, as Kathy showed from Kitty's work, it's never a single organism, except in the most extreme temperatures. It's always consortia. So for us, it's a real chance to say that this stromatolite, 3 billion years old, was made by cyanobacteria. Of course, there's 10,000 different types of microbes in modern stromatolites. So, I mean, that's just some of the complexity that we face going back in the record.
I think my concern though is to throw out the textural, the pictures I showed you are what allow us to hone in on the places where we most would expect there to be life rather than just looking anywhere. And so you've just got to be very clear that yes, we understand that there are pseudo fossils out there, but also don't throw out the potential for these to be fossils just because you don't have the organic molecules preserved, because that's an issue for sure that we're facing all the time with our work. That's why the instruments that go to Mars are all about measuring organic geochemistry and less so about, until recently, about collecting samples or other things that we might do ourselves.
Okay,
Right.
I think we should stop the official part there. I'll stop the recording and thank everybody for coming out. And if anybody wants to stand around and have a chat with Kathy, we'll do that. Thanks, Anna. And yeah, we'll see you all next time. Thanks a lot.
Thank you. Nice to